Okay.
Is anybody else on that?
Yeah, there's like 5 or 6 people on there. I am not muted at the moment. No, I'm not. Do you want to play your music?
Yeah.
Okay. Hi, everyone. We're still a couple minutes away from starting. I'm just playing some random background slides, but I don't have any sound to go with it. So I just wanted to say that Cindy's going to play a little music, I think, for us. And we're going to start in just a few minutes, but we're going to give people plenty of time to get on and to get the technology working. We should be both live now on Zoom and on YouTube. It's just on my channel, Department Chair's channel, Jill Fenton, if you want to direct family members there. You know what? Shit. I can't do it while I'm-
Jill, we're not seeing it on the YouTube.
You're not seeing it on the YouTube.
It's 20 seconds behind.
Can you see anything on this? It's 20. Is it 20 seconds behind? I can
Oh, hold on, let me refresh and see.
refresh.
'Cause
and
I just
see.
see, I have my group Slack popping in saying it's not up, but let me just.
It's not up
refresh.
on the YouTube yet.
Hold on, let me.
It
refresh.
tells me yeah, no, I want to know that if we're not. I can't watch both on my
Yeah,
computer.
they're on. It's
Okay.
on. Okay.
The YouTube one's a little
Delay. It
behind.
was the delay. All right.
Yeah, the YouTube is about 20 seconds delay. But unfortunately, it echoes if I have them both on the same computer. So
Yeah.
I don't like to echo myself.
My bad.
Again, we'll start in just another minute. I'm just going to give everybody a chance to get their technology working and to get online. Okay, we're going to start in just a moment. Ooh, somebody's on. I'm waiting for Cindy to join the call so I can enable her to share her screen. When I can't make her a host until I can enable her to share her screen. But anyways, welcome everybody to our live ceremony for our graduate students for the chemistry department. My name is Jill Benton and I am the chair of the chemistry department. Just a little bit of housekeeping as we start today. In the name of full disclosure, I want to remind you that we are both recording this on Zoom and broadcasting live to YouTube. So there's absolutely no thought of any privacy. The whole ceremony is public. And again, if your family wants to join in the live ceremony, any friends, they can just search for Jill Benton on YouTube and it's streaming live there on my channel. If you don't catch us live, or if you want to rewatch today's events as many times as you want to, the video from this live broadcast will be on the Jill Benton YouTube channel for you to enjoy later. Okay, so as we begin today with our formal ceremony, before we kind of begin our formal ceremony, what I want to do is have everybody for a moment, if you're a graduate or if you're a faculty member, can you go ahead and turn your camera on for just a minute? Because what I want to do is I want to take a moment. We went live today for a reason. We didn't want to just do a video recording. We went live so we could see each other. So I want to start this ceremony by having you spend just a moment, you put it on gallery view for a moment on your YouTube and go ahead and scan through and see your friends. You can wave hi, et cetera, et cetera. See your friends, see your faculty members. We're really proud of you and we're all here to celebrate you today. Nobody gets through a chemistry degree by themselves, right? All of you started this journey out and you worked in lab groups, working with teams of people. You've been TAs, you've made lots of friends along the way. That's one thing that I'm always impressed when I go to like PhD defenses, right? How people thank their friends. They made lifelong friends. So I just wanted to spend a moment where we saw each other for a minute, saying hi to each other, that we didn't get to end maybe quite the way we thought we would for a lot of you for your graduate career, but I wanted to make sure that you saw the people that make up kind of the UVA community and the graduate student class. I also want to just take a minute to invite you to turn, if you're with your family today, and say thank you. If you're not with your family, call your parents, your family, your friends later on today. Maybe they're watching. Hi, moms and dads. Actually, one of the cool things about this format is that I can say hi to moms and dads all around the world. That we have a lot of international students who maybe typically it would be out of, we wouldn't be able to, invite a parent here, but they can watch in this format. So that's kind of cool. But, again, we've been in strange times lately with the pandemic upon us, but we all know that family and friends are extremely important. Again, you don't get here finishing your graduate degree without a lot of family and friend support. So thank you to all the families and friends. If they're with you, turn and say hi and thanks. We want to make sure that we include them as well. Okay, so if you don't want to leave your video on, you don't have to. We're a small enough ceremony that that's a wonderful thing. If you want to leave it on, you can. One of the things that we were able to do during the undergrad ceremonies, a lot of people chatted congratulations. We're going to give a time at the end where you can speak to each other, you can chat at each other, et cetera, et cetera. But if you want to chat congratulations to each other during the ceremony as well, you can feel free to do that. Again, we're live so we can interact. And so feel free to use the chat function if you want to. Okay, well, I just want to say a few things before we begin today. That again, we are virtual today because we wanted to honor you. A reminder that the university is planning to hold in-person final exercises for you. They've set a hopeful date of October 10th, 2020. But if that date doesn't work out, if they don't feel it's safe, then the backup date is May 29th. 2021. So we do hope that we get to honor you in person, but I don't know how feasible that will be for some of you. So we wanted to make sure we would mark the occasion today. But if you didn't know those dates, go ahead and mark them on your calendars. Okay, so today we're here to honor you, right? Things may not have ended quite as we expected this semester, but we really want the focus to be on you and your achievements. We're really proud of all that you've done to achieve your goals and receive your graduate chemistry degree. A degree in chemistry is not just an acknowledgement that you know some science. It's an acknowledgement that you can persevere through some of the hardest classes and, quite frankly, hardest research in the university. Graduate school especially immerses you in the process of research, which we all know can be frustrating. Families, you probably already figured this out, but research doesn't always work. And so these students have really persevered through the last few years, right, in order to be able to achieve, in order to learn something new, create new knowledge in science, and on their kind of quest to learn chemistry. Along the way, most of you likely learned to teach. Many of you served as TAs. And quite frankly, this is also a great life skill, working with different types of people, training that will be invaluable as you move on to your next pursuits. We know that people in our department move on to a variety of things from graduate school. Some of you might be going into industry, pharmaceutical, chemical, that kind of thing. Others of you might be going into teaching careers and academics. Others go into something like sales or the military or all kinds of things. And it really doesn't matter what you go into. We know that what's really important are the skills that you've developed in critical thinking and problem solving, communication. Those are the sorts of things that are going to really be with you throughout your career. And obviously in this time of kind of a global pandemic, we've seen just how much we need scientists. We need scientists to address the medical questions of today, right? Drug discovery, vaccine development, basic mechanisms of disease, right? All of these are things that chemistry graduates such as you have the skills now to tackle. But those aren't, of course, all the only societal problems that we have. Those might be what we're concentrating on today, but there's so many other things, right, that we need good scientists to be able to do. Things like alternative energy resources or smart materials are challenges that are there for the future as well. And then one of the things that I always like to mention is that some of you might have spent the last few years studying things that the society would consider pretty esoteric, maybe even not real useful, astrochemistry. But one of the best things I think about being a chemist is just that we don't always have to be inherently useful. The process of discovery is in and of itself a good pursuit that humans, I think, have a fundamental desire to understand how the world works around us. And hopefully you've been able to spend a few years really digging deep kind of in that quest for knowledge. So today we are here to celebrate your accomplishments, to tell you that we're proud of you and all that you've done in the last few years. We know that you're going to go on to great things. You've developed very good chemistry skills, but quite frankly, much resilience as well. So as you go on, we just ask you to please keep in touch. Let us know how you're doing on your journey. We love to hear from all of our alumni, our chemistry who's, and we want you to remember that you'll always be a part of our UVA chemistry family. So I'm going to just ask Brent Gano now, our Director of Graduate Studies, just to give a brief set of congratulations to our graduates.
Okay, thank you very much, Jill. I would also like to welcome all of the graduating students along with their family members, friends, and colleagues from UVA and beyond. As the Director of Graduate Studies for the Department of Chemistry, it is indeed an extreme pleasure to help celebrate the tremendous accomplishments of this group of scholars. Obtaining an advanced degree in chemistry is intellectually challenging and rigorous. For all the graduating students, you should take great pride in reaching this point. I'm going to start out with a round of applause at the beginning. And for all of the family members and friends whose support has provided them the foundation to be successful, we thank you as well. Graduates, you will now move on to lead new research focused on medical technologies, more environmentally benign plastics, new energy advancements. You will become patent lawyers or patent examiners. You will become great educators. You will inform public policy and many other important endeavors. And now that you are graduating, we look forward to watching your coming professional as well as personal accomplishments. Your faculty advisors and teachers have watched you struggle, learn, and excel. And as we have worked with you, we have established very close relationships. So as Jill noted, after today's celebration, we certainly hope that you'll keep us updated and stop by to visit us when you have an opportunity. Again, congratulations and enjoy today's ceremony and the celebrations that will follow it.
All right, thanks, Professor Gano, for that. So now we're on to the main part of the ceremony. Again, our main focus today is to celebrate you. Our Associate Chair, Professor Charles Grisham, is going to read the names of all the graduates. You'll see a picture come across the screen if you sent us one. Some people sent us one, some people did not. When we get to the PhD students, we are going to see if we can ask their faculty mentor, if they're on the call today, to say a word about them and their research. So we'll see how that works, but we think it'll be good and interactive. And then at the end, again, I promise we'll give a chance for everybody to unmute and give a big cheer for all of the graduates. So with that, I'm going to turn it over to Professor Grisham, and I think Cindy is in the background as always, and she's going to share the screen.
Okay. Hello, everyone, and congratulations to all the graduates. We start with Bushra Abdelmalik, Master of Arts in Chemistry. Matthew DeSelm, Master of Arts in Chemistry under the direction of Professor Rob Garad. Hongying Dong, Master of Arts in Chemistry. Evan Hall, Master of Arts in Chemistry under the direction of Professor Brent Gano. Dijia Kong, Master of Arts in Chemistry. Mary Rad Morris, Master of Arts in Chemistry under the direction of Professor Kevin Lehman. Emmett Kellum Pert, Master of Arts in Chemistry. Jordan Nathaniel Rogers, Master of Arts in Chemistry under the direction of Professor Don Hunt. Wyatt James Summerlin Smith, Master of Arts in Chemistry under the direction of Professor Robert Gilliard. Jillian Turner, Master of Arts in Chemistry under the direction of Professor Charles Mahan. Junlin Wu, Master of Arts in Chemistry under the direction of Professor Sen Zhang. Han Jung Kim, Master of Science in Chemistry. Title of the dissertation is Investigation of Flavinium Salts as Catalysts for Intermolecular CSP3H Amination under the direction of Professor Mike Hilinski. Lauren Leesky, Master of Science in Chemistry. Title of the dissertation is Transition Metal-Based Complexes and Organic Small Molecules for the Electrochemical Reduction of Carbon Dioxide and Dioxygen under the direction of Professor Charles Mahan. Amanda Mattel, Master of Science in Chemistry. Title of the dissertation is Targeting Mutant TERT Promoter in Glioblastoma to Inhibit Replicative Immortality under the direction of Professor John Bushwelling. Maura Belanger, Doctor of Philosophy and Chemistry with a concentration in biological chemistry, recipient of the Immunology Training Grant Award. Title of her dissertation is Development and Characterization of Analytical Tools to Study Immunity ex vivo under the direction of Professor Rebecca Pompano. And I believe Professor Pompano is with us and would like to say a few words about Maura.
Yes. Let's see. All right. So I just wanted to be able to introduce Maura. So Maura's PhD is extra special because she is the first PhD student to graduate from my laboratory. And what that really means is that she joined as a fearless first year, a lab that was not even set up yet, and then helped establish all of the fundamental protocols that are now used routinely in our research. And Maura has been really instrumental in not only laying the scientific foundations, but also in training and ensuring the rigor of all of our subsequent students. So Maura's research, as you heard from the title, really established a new system to study immunity outside of the body, and now enables other students in the laboratory to analyze the chemistry of how cells signal to each other within that tissue. And Maura is the first person in the world to have delved into such depth into how we can keep these tissues in a state where we can really see what's happening with the cells. Her work has led to 4 papers on which she was co-author or lead author, and she was awarded a competitive immunology training grant fellowship during her time. So I'm very proud of Maura. Congratulations.
Thank you, Rebecca. Jeffrey Brulé, Doctor of Philosophy and Chemistry with a concentration in organic chemistry. Title of the dissertation is Sulfur Triazole Exchange Chemistry for the Global Targeting and Liganding of Functional Tyrosine Sites, under the direction of Professor Ken Su. And I think Ken is with us. And Ken, would you like to say a few words about Jeffrey?
Oh, yes, absolutely. So it's really a great pleasure to be able to introduce Jeff. Jeff was among the group of one of my first graduate students. So really holds a special place, in our relationship and in the history of the lab. So, and I remember Jeff joining my group and expressing his interest to develop new chemistry to study protein biology. And, I would say he's definitely accomplished that. He was one of the key members that helped establish sulfonyl triazoles as a new electrophile that has some broad applications in protein biology and drug discovery. Jeff was, Jeff was, one of the most hardworking, most reliable students I've ever had the pleasure of working with. I'm going to really miss him, and I'm sure the lab is really going to miss him. And yeah, he was always a leader and really helped train a lot of the new generation of students in my group. So thank you for all you did, Jeff. And yeah, waving back. And yeah, looking forward to the fall when we get an actual in-person and I get to do this again. So anyways, good, I know you have some options and everything, so good luck with everything. And again, thanks for letting me introduce you.
Thank you, Ken. And on to Charlie Clark, Doctor of Philosophy and Chemistry with a concentration in biological chemistry, recipient of the Department of Chemistry Outstanding Graduate Teaching Assistant Award, and also the Eric Buell Memorial Award. And he placed first in the 3-minute thesis competition in 2018, also received the SciEx Young Scientist Award and the Dr. Frank Finger Graduate Fellowship for Teaching Award, and finally the Jefferson Scholars Foundation Dissertation Year Fellowship Award. The title of the dissertation is Microfluidic Applications in Forensic Science, and he did this work under the direction of Professor James Landers. James, if you're with us, would you like to say a few words about Charlie?
I don't think he was able to join today.
Yeah, and I don't think he was. Well, I know he would say some nice things about Charlie, and we're going to miss you. I'll just say that. How's that? Thanks, Charlie. Logan Aaron Comby, Doctor of Philosophy and Chemistry with a concentration in organic chemistry. Title of the dissertation is New Methods Toward Efficient Catalytic CO and CN Bond-Forming Reactions. And he did this work under the direction of Professor Mike Helinski. Mike, would you like to say a few words about Logan?
Yeah, I can just say a few words briefly. Logan, sad to see him go, even though he's been gone for a few months ago, and I'll talk about that in just a moment. But Logan was the second PhD to graduate from my laboratory. So he really was among that group of people who really got the laboratory's research started. You know, one of the most hardworking and most self-motivated students I've had yet during that time in those early years. He contributed to a number of different projects. I think he had 4 or 5 publications out of my laboratory, but I'll just briefly mention 2. He was very a key laboratory member in establishing our organocatalytic CH amination reaction, which as one of the results we've become most known for in the intervening years. And a second project that he contributed very in a key way to was developing some of our new synthetic methodology towards the development of heterocycles, which was sprung out of basically his own serendipitous discovery and his motivation to follow up on that. And I'm lucky that that he had that motivation, because then that led to a whole new, now federally funded project in my laboratory. So I miss him quite a bit. He is now, it's been several months into a postdoc at the University of Michigan, where he's doing quite well, so I hear. And so congratulations, Logan.
Thank you, Mike. Stephen Deckermanji, Doctor of Philosophy and Chemistry with a concentration in inorganic chemistry, recipient of the Department of Chemistry Outstanding Graduate Teaching Assistant Award. The title of his dissertation is Applications of Redox Processes to Molybdenum and Tungsten Dearomatization Agents. And he did this work under the direction of Professor Dean Harmon. Dean, would you like to say a few words about Steve?
Yes, thank you, Charlie. Hello, everyone. So our research group aims to develop new methods for synthesis of molecules that are designed to be ultimately new medicines. We do this through the use of molecules that act a little bit like a teeny vise, which can clamp onto a particular section of the molecule and operate on it. Sometimes that vise, which includes either tungsten or molybdenum, clamps down on the wrong part of the molecule. Stephen's project was to figure out how to loosen its grip just enough that it could slide over to the correct target. He also learned how to make this vise let go completely, and this allowed the molecule to move on to the next target. Stephen has published already 6 manuscripts in ACS journals, including a manuscript in JACS. Very hardworking, and his project really took us in completely unexpected and new directions, and I really, really enjoyed working with him. Congratulations, Stephen.
Thank you, Dean. We're going to move on now to 3 students from Professor Hunt's lab. Cindy, if you can back up one. There we go. We have 3 students in a row from the Hunt lab, and I believe Professor Hunt wants to make some comments at the end of the introduction of all 3. So we'll start with Robert DiPolito, Doctor of Philosophy and Chemistry with a concentration in analytical chemistry, recipient of the Jefferson Scholars Foundation Research Award. The title of his dissertation is Analysis of Protamines and the Investigation of Concentration Effects of an Immobilized Aspergillopepsin-1 Reactor. That was done under the direction of Don Hunt, of course. Next is Elizabeth Ducelis, Doctor of Philosophy and Chemistry with a concentration in physical chemistry. Title of her dissertation is Ion-ion Reactions in Primary Structure Interrogation of Intact Proteins by Mass Spectrometry, of course, under the direction of Professor Hunt. And finally, Joshua David Hinkle, Doctor of Philosophy and Chemistry with a concentration in biological chemistry. Title of his dissertation is Antibody Sequence Analysis by Controlled Proteolysis and Ion-ion Chemistry. And now I think Don would like to say a few words about each of these students.
Hi, thanks very much for letting me say a few words. I'm going to begin by telling you that the emphasis on all 3 of these PhDs was to understand and to be able to characterize antibodies. And if you look at my picture, I'm going to make an antibody, because an antibody looks like a lobster with a long tail, and then there's 2 claws, and the 2 claws combined to something in a fish or something that they want to eat. antibodies do the same thing. And they have basically 50 amino acids making up the building block, making up the size of this chain, and then another 250 up here making the claw, okay? And if you've been reading the newspapers about this virus that's going around, then you know that everybody hopes they have antibodies to protect you from this virus, because an antibody is something that's produced by your immune system. And you need it pretty badly. So the question is, can you characterize them and make new ones, okay? And so Elizabeth was the first student to put on the project, and her challenge was to build a mass spectrometer. That's the technique we use. I'll say a little about that in a minute. But this mass spectrometer stuff these instruments cost about $1 million. They operate under high vacuum. They got a lot of electronics and wave functions, and they also have chemistry happening. So her job was to modify these instruments so you could characterize the structure of antibodies. So the first thing she had to do, which was not easy, was to modify the hardware. She had to modify the electronics and the wave functions used in these instruments, and she had to develop new chemistry. We do chemistry inside the high vacuum of a mass spectrometer. And so it's pretty amazing sensitivity. If you can imagine what one crystal of salt looks like, a tiny little crystal of salt, what we're playing with is 10 billion times less than that to put into this instrument and come out with a complete structure of an antibody. So after she had developed new chemistry, new reagents, new electronics and wave functions, she started by applying it to normal proteins. They work fine. And in fact, she completed the sequence of a hemoglobin molecule. It got sent to us from California because they couldn't figure out what it was. She figured it out. And because she figured it out and got it correct, we now have a hemoglobin molecule called hemoglobin Charlottesville. So she has one named for her. Okay, so that's Elizabeth. I'll come back to her in just a minute, but the next one is Josh Hinkle. Josh Hinkle followed on. He was there at the same time, and of course worked somewhat with her and on his own. And his challenge was to, after Elizabeth made the instrument work, he used the instrument and combined it with a proteolytic enzyme, which would chop up the protein into big pieces. So we use it as a scissor. And he was the first to apply this technology of an immobilized proteolytic enzyme. And he generated a near complete structure of an antibody, and it actually took him only about an hour and a half. So that is unbelievable. So that is his major accomplishment. And then following along, Robert DiPolito, who was also in the lab. He comes a little bit later. But his collaboration, or his focus, was on this enzyme system. And he actually made an amazing discovery that it now means you can take this incredible small amount of material, pass it through this enzyme degradation system, and you can characterize not only just antibodies, where the normal ones have two claws like a lobster. If you have bispecific antibodies, then you make an antibody on this side and you make a different antibody on that side. And what it does is allow you to bind to a cell here, bind to a cell here, and bring them into close proximity. And so that is a huge industry in the biophysical community right now. They're called bispecific antibodies. So that doubles and triples your problem to characterize them. And Rob DiPolito figured that out and fully characterized the first bispecific antibody. So amazing accomplishments from all three. And congratulations to Elizabeth, congratulations to Joshua, and my congratulations to Rob DiPolito as well. They were amazing students. Thank you.
Thank you, Don. Dominique Maurice Mafucci, Doctor of Philosophy and Chemistry with a concentration in Physical Chemistry. Title of the dissertation is Methods of Astrochemical Modeling, a Multidimensional Approach. And this was done under the direction of Professor Eric Herbst. And I think Eric is with us today. And Eric, would you like to say a few words about Dominique? Perhaps not. Well, in any case, we'll move on. Pumadate Puttankam, Doctor of Philosophy and Chemistry with a concentration in Analytical Chemistry, recipient of the Sidney Hecht Graduate Fellowship Award, The title of his dissertation is Novel Microelectrodes and Methods for Real-Time Electrochemical Detection of Neurotransmitters, under the direction of Professor Jill Venton. And I know Jill is here and would like to say a few words.
Yes, thank you. It's my pleasure today to introduce you to Max, who's really been a jack of all trades in the kinds of projects he's been able to take on in my lab. So my lab generally tries to develop sensors that we can put in the brain to find out more about how your brain chemistry works. And Max really worked on a variety of projects. One of his projects he worked on was just fundamental electrochemistry so that we could understand the electrochemistry of the molecule histamine. You sometimes take antihistamines, right, if you have allergy problems, especially now in the spring. But nobody really understood the electrochemistry. It's hard to believe. And so he was able to come up with a fundamental mechanism for histamine electrochemistry. And that means now that we can take that mechanism and apply it, hopefully, to being able to make measurements of histamine as a neurochemical in the brain. He also worked on the materials side of things, developing new materials for microelectrodes, including things like nanodiamonds and carbon nanohorns. And so he did a lot of work in trying to craft better kind of electrode surfaces to be able to look at neurotransmitters. And then the last thing he did, as I said, in the jack of all trades, is that he wrote computer algorithms. So when you go to the brain and you put an electrode in and you try and measure neurochemicals, of course, you need to be able to figure out what is the neurochemical that you want to look at and what is noise, and be able to do that well. And so he worked in a collaboration with Professor Gallman's group to use image analysis in order to be able to really pick out different neurotransmitters from all of our data. We get hours and hours of data and be able to identify when we see different neurochemicals. So we're really proud of Max. He came to us from Thailand, and he's going back to Thailand this summer. He's going to become a professor at a university with a really long name, but it starts with Chula, and he tells me that's just the nickname. So we're gonna go with that. So he'll be a professor back in Thailand, and we're looking forward to continuing our collaborations with him and wish him the best in his career. Thanks.
Thank you, Jill. Next is Julian Roca, Doctor of Philosophy and Chemistry with a concentration in physical chemistry. Title of the dissertation is Dynamic Assembly of the Type 3 Secretion System in Yersinia enterocolytica, probed by super resolution fluorescence imaging. And that was done under the direction of Professor Andreas Gallman. Andreas, would you like to say a few words?
Yes, thank you, Charlie. Julian Roca was also the first student in my lab and to graduate from my lab. When he joined, my lab was empty of equipment and empty of personnel. He then built a microscope to watch single protein molecules move around in living bacterial cells. Next, he wrote the analysis software to resolve the different motion behaviors of the proteins that he was following. And then finally, he applied these experimental and these computational tools that he built to further our understanding of how bacterial pathogens secrete toxins into host cells during an infection. It has been a true pleasure and the privilege to watch him complete this entire process from the beginning to the end. And so thank you, Julian, for your hard work and for your accomplishments. Best of luck in your new job and congratulations.
Thank you, Andreas. Jacob Smith, Doctor of Philosophy and Chemistry with a concentration in inorganic chemistry, recipient of the Alan Gwathmey Award for Best Paper in the Physical Sciences. The title of his dissertation is Strategies Towards Nucleophilic Additions to Eta-2-Bound Arenes. This was done under the direction of Professor Dean Harmon. Dean, would you like to say a few words about Jacob?
Yes, thank you, Charlie. So remember that molecular vise I told you about? Well, Jacob's PhD work involved using that vise to create new molecules that never existed before. Now, when a person ingests a medicine, that medicine's broken down in the body, similar to how the body digests food. This means that a person needs more of the medicine and higher doses, and that can lead to unintended side effects. Jacobs learned how to replace certain hydrogen atoms in a molecule with a close relative called deuterium. The deuterium acts almost exactly like hydrogen, but it forms bonds with carbon that are just a little stronger. This can slow down the rate at which the medicine breaks down and can alter what it breaks down into. That would be lower doses and less side effects. Jacob's work will be described soon in an article that comes out in Nature that's published next week. Congratulations, Jacob, for superb work.
Thank you, Dean. Ellen Spears, Doctor of Philosophy and Chemistry with a concentration in biological chemistry. Title of her dissertation is Mass Spectrometry Characterization of Post-Translational Modifications on the Arabidopsis Growth Regulator Protein RGA. And this was done under the direction of Professor Don Hunt. Don, would you like to comment on Ellen?
Sure. So this is sort of another very exciting project that Ellen is one of the people working on. It has to do with the Green Revolution. And this is basically back in the early parts of the century, mid-century, people actually tried to induce well, they were afraid that there wasn't going to be enough food for the number of people that were being born on the earth. And so what they did was mutate all the proteins that were being, like in corn plants and rice and so forth. And they eventually produced semi-dwarf plants, which were much more sturdy than the large intact plants. And these dwarf plants were more productive than the original plant. And so now we don't have to worry about not having enough food. And Norman Borlaug got the Nobel Prize in 1970 for his development of these semi-dwarf mutants. It was done in wheat, it's done in maize, and it was done in rice. So pretty amazing development. But even though they made it happen, the reason and the logic and the biochemistry underneath it is not well known. And so Ellen and several other members of the group are now collaborating with plant biologists to fully characterize the plant proteins that are involved in this process. And it turns out that it has to do with cell signaling, and cell signaling usually means post-translational modifications on plants, and those modifications come on and off, and you turn on the plant or turn off the plant. So a full understanding of these cell signaling events is likely to lead to even better plants in the future. And Ellen was one of the first members of the group to participate in that, and she was very productive. So congratulations to Ellen for sure.
Thank you very much, Don. Nicole Swope, Doctor of Philosophy and Chemistry with a concentration in biological chemistry, recipient of the NSF Graduate Student Research Fellowship Award. Title of her dissertation is Spectroscopic Studies of Glycosylated CCAM1 ectodomains and Membrane Protein Folding in Lipid Detergent Mixtures. This was done under the direction of Professor Linda Columbus. Linda, would you like to say a few words about Nicole?
Absolutely. So Nicole has been a fantastic graduate student in the lab, and she already came in so strong. To add to her accolades, she also was on the biotechnology training grant. She did a externship at MedImmune and has a publication from that. She is mentored in undergrad on a totally different topic to publication as well. And one of my proudest moments was, and besides today, was at a Gordon conference. Nicole actually had a talk on the first day after the keynote speakers. And I couldn't come, I was getting there that night. And she rocked it. And I got to spend 5 days in pure proud state of hearing about how fantastic Nicole was throughout this conference. And for those of people that aren't familiar with Gordon conferences, everybody's there. There's usually a Nobel laureate there. It's in your field, and it's a very prestigious thing. And so it's a very proud moment. So Nicole's topic, her titles are very long because she worked on many different projects, but all related to membrane proteins and their environment. And so in a cell, if you think of the room that you're in, the membrane is the walls and the membrane proteins are the doors and the windows. And so we're interested on what do the doors and windows look like and how do they move to carry out their function. And just to bring you to the most relevant types of membrane proteins, COVID, the coronavirus actually gains entry into our cells through ACE2, which is a membrane protein. And so understanding these proteins is extremely important. We really don't understand the fundamental driving forces of how those proteins move and how they interact with the walls. So how does the door move and how does the wall help facilitate that? And so it is with great pleasure to congratulate you, Nicole. Way to go, Dr. Swope.
Thank you, Linda. Justin Wild, Doctor of Philosophy and Chemistry, with a concentration in organic chemistry. Title of the dissertation is Synthetic Applications of Molybdenum and Tungsten Dearomatization Agents. This was done under the direction of none other than Professor Dean Harmon. Dean, would you like to comment on Justin?
Thank you, Charlie. Justin also uses those molecular vices I've been talking about, which in our group we call dearomatization agents. That's their fancy name. But he uses these to make new molecules that, again, we hope will lead to new medicines. Justin's particular expertise comes from a molecule called pyridine, which is found in crude oil. And with a very clever series of operations, Justin's figured out how to carry out with this vice. He converts this pyridine into new types of molecules that are going to be evaluated by the drug company Lilly through a material transfer agreement with them to study their efficacy in treating various diseases. Justin's an extraordinarily hard worker and has made just a tremendous amount of progress in the short time that he's been with us. He's got 6 publications in ACS journals, 2 more on the way. Justin, congratulations for a fine, fine job.
Thank you, Dean. Next is Shen Zhang, Doctor of Philosophy and Chemistry with a concentration in analytical chemistry. Title of the dissertation is Genetically Encoded Fluorescent Biosensors to Light Up Biological Signaling. And this was done under the direction of Professor Huiwang Ai. Professor Ai, are you here and would you like to comment on Shen?
Yes, thank you. Thanks for the opportunity to let me to speak a few words on Shen. Shen actually joined my group when I was at UC Riverside before I moved to University of Virginia. And about 3 years ago, we relocated to University of Virginia. He came to UVA with me with the lab actually. He worked in my lab for about 6 years. That's really, it's a long journey, but he's talented. His persistence eventually has led to a good outcome, I would say. In his first project, he developed biosensors to image zinc in lab cells and organisms, and very innovatively, he applied this biosensor to study beta cell function. Everybody, including us, we have beta cells in our pancreas in response to glucose, such as we eat a meal, and beta cell we are secrete insulin. And Shen's sensor actually can use to evaluate beta cell function. And for diabetic patients, there is a investigational therapy, which is doctors try to transplant beta cells from deceased donors to type 1 diabetic patient. And the Shen's sensor can sense beta cell function. So right now, this sensor actually is being used by doctors at the UVA Medical Center. So we are trying to advance this novel experimental therapy. In another project, Shen developed a genetically encoded sensor for serotonin, which is a molecule in our brain, which is important for our happiness. We want to have more serotonin in general to be happier. And Shen's work actually enabled the imaging of serotonin in live animals. And basically this will pave the road for further study about serotonin and behavior with animal models. And in addition, Shen also developed a general method to expand the colors of fluorescent biosensors, also contributed to the development of multiple biosensors for hydrogen sulfide, which is a gas molecule also used as a signal transmitter in our bodies. Shane has been very productive. He has been the first author and co-author of 6 manuscripts, including one paper actually will be published on Nature Chemical Biology, and with another paper is being reviewed by Nature Methods. And congratulations, Shane, for this well-deserved degree. And I wish you great luck and have an excellent future. Actually, thank you.
Thank you, Hui Wang. Meng Zhuang, Doctor of Philosophy and Chemistry, with a concentration in analytical chemistry, recipient of the Presidential Fellowship for Collaborative Neuroscience Award. Title of the dissertation, Boron Dipolymer Materials for Oxygen Sensing and Imaging. This was done under the direction of Professor Cassandra Frazier. Professor Frazier, if you're with us, would you like to say a few words about Meng?
Yes, thanks, Charlie. Yes, Meng's done a great job, and her research is on luminescent biomaterials for imaging and sensing. And sometimes the materials just have all kinds of beautiful colors and allow you to image different parts of cells or tissues or in vivo in organisms. But one really special property in the materials that Meng helped to develop and optimize is that these materials allow for oxygen imaging. And that's very important in different branches of medicine. And consequently, Meng has had the opportunity to collaborate with people in a variety of different fields. And so these imaging agents Meng has made have been used in tumor imaging and wound imaging, brain imaging with Professor Kapoor, who's the head of the Brain Institute at UVA. So Meng has had the chance to work from materials all the way to doing biological experiments in the brain. And she's also collaborated with the Pompano lab and with Maura, who's also graduating this year. They have a joint publication. And Meng has just had made many contributions in adapting these luminescent materials, these glowing materials for all these many different biological applications. And I just want to congratulate her on a successful PhD.
Thank you, Professor Frazier. And we close with 2 students from Lin Pu's lab. And we took them out of alphabetical order, but we are going to present them to you now so that he could provide a couple of videos for these 2 students. The first one is Gengyu Du. We'll wait for Cindy to share the screen here. Cindy, are you there?
I'm here. Jill has the videos.
Oh, but we need the slides first.
Oh, my bad.
That's okay.
Yeah, let's show their picture first, then we'll do the videos.
That's right. Slide first, then video. There we go.
Sorry.
No problem. All good. Gengyu Du, PhD in chemistry, Doctor of Philosophy and Chemistry with a concentration in organic chemistry. Title of the dissertation is Fluorescent Recognition of Amino Acids, a Micelle Approach. And this work was done under the direction of Professor Lin Pu. And we'll do the next one as well. Judan Wu, Doctor of Philosophy and Chemistry. Title of the dissertation is Fluoresce Phase-Based Enantioselective Fluorescent Screening for Asymmetric Catalytic Reaction and Enantioselective Fluorescent Recognition of Amino Acids, also under the direction of Professor Pu. And now we can turn to the videos.
Every years at UVA, she then conduct research in the area of asymmetric catalysis and enantioselective fluorescent recognition. He discovered that a fluorescence-based fluorescent probe can be used to conduct a rapid screening of chirocations for the asymmetric amino alcohols. And this work provides a potentially useful method for high throughput screening of asymmetric reactions. In another work, he discovered for the first time that a fluorescent probe can be used to determine both the concentration and the enantiomer composition by excitation at two different wavelengths. This method can significantly simplify the analysis of chiral substrates. So Shida has made important contributions in his PhD study and is an excellent student. Congratulations again to Shida. Congratulations to Gingyue for receiving the PhD degree. Gingyue came to UVA from University of Science and Technology of China with a bachelor degree. In the past several years at UVA, he has worked on the development of fluorescent sensors for the enantioselective recognition of amino acid. He has discovered that using Brachypolymer-based micelles not only allows the water-insoluble fluorescent probe to be used in aqueous solution, but also shows greatly enhanced selectivity and sensitivity. His innovative work has pioneered the use of micelles in enantioselective fluorescent recognition of chiral compounds, and this should contribute to further progress on this area. Gingyue is an excellent student, has done important research work in his PhD study. Again, congratulations to Gingyue. Congratulations.
And I think that concludes my part. So back to you, Jill.
All right. Well, thank you to everybody for being here today. I said that we would take a moment at the end. We're going to unmute everybody, and we're going to allow you to give everybody a big round of applause on all of your accomplishments. So congratulations to everybody.
Congratulations, everyone.
Yes, so that concludes our formal ceremony for today. Again, I would like to thank Cindy Knight for all of her work in helping to put this ceremony together. Really, she does amazing work with all the photos and videos and all of that. And to our graduates, we want to say again that we are proud of you and all of your accomplishments. Please keep in touch. You'll always be a part of our UVA chemistry family. And so I encourage you to celebrate safely. I'm going to leave this Zoom on for just a couple more minutes. Or say hello to somebody. Go ahead and do that. And again, congratulations to everybody and stay safe. For today, again, I would like to thank Cindy.
Congratulations, everyone.
Congratulations, everybody.
Congratulations, all of you. We're really proud of you.
Good job, folks.
Congratulations. Even though I don't work with you guys as much, you're always a part of our family. I wish you all the best. Please keep in touch.
Use that doctor well. Bye, guys.
Congratulations. Best of luck in the future.
Congratulations, everyone. Get used to, you'll have fun getting used to having doctor in front of your name. It'll feel strange at first, but it's a very big, proud accomplishment.
All right. Well, it looks like most people are kind of signing off. Again, thank you to everyone. Congratulations, and celebrate safely, everybody. Thanks for joining us today. And again, you can watch this all again on YouTube if you didn't catch it live. Thanks.